My first section, the bilayer, membrane structure, peptidins, and the meaning for messaging. It is difficult to find a suitable entry point when you dedicate yourself to a specific perspective, looking into a cell and finding such a complex, intelligent design. It's such a fascinating topic that I could rave about it for hours. I don't want to get too much in the details in this presentation, still hoping I can hopefully bring a basic problem of the LNP a bit closer to you. Nevertheless, I have to go into some very technical details, as unfortunately there seems to be a very widespread misunderstanding about the complexity of eukaryotic cells. Many people believe that you simply put something into the cell and then the cell does what it does without taking into account how complex the regulation of each individual process really is. Since my perspective is focused on the LNPs and the damage potential, I must first deal briefly with the phospholipid bilayer, in other words, the cell membrane. If you take a closer look at it, you quickly realize how complex its structure is. Imagine a three-dimensional mosaic of countless lipids that are in permanent oscillation due to forces such as van der Waals forces. The membrane is divided into two lipid layers, which are anionical charged, which means negatively charged. The lipid heads are hydrophilic, which means they love to interact with water, while the saturated and unsaturated fatty acid tails of the lipids are hydrophobic, which means they don't react with water. A kind of equilibrium prevails in the hydrophobic center of the bilayer, and I can only ask you to keep this point in mind, as it will play an important role in my discussion at the end. In addition to the normal lipids, we also have sphingolipids, which are made up of completely different components. In the hydrophobic center, we also find cholesterols. But we have something else, and this is where it is getting really tricky. We also have the so-called PIPs, phosphoinositoids, which are formed from the substrate phosphatidylinositols in binding with phosphorus, which make up about 1% of the membrane. And they are the main drivers in endocytic processes for secondary messaging, which I will explain in the next section. Endocytosis refers to the uptake of exogenous material into the cell, which we call internalization by invagination and constriction of parts of the cell membrane with the formation of vesicles or vacuoles. There are different types of endocytosis. For example, pinocytosis, which means endocytosis of liquid particles, and phagocytosis, which means endocytosis of large solid particles. The head of the pips shows exclusively into the cytosolic direction of the cell. So, said with that, and showing the complexity of the membrane, I would like to make one particular point. If we want to understand transfection, in other words, a liposome, or in our case a much more complex technology of lipid nanoparticles, a composition of individual lipids, cutting into the membrane, or you can say penetrating it, we must first understand the functions of the membrane as far as we can. This results from the fact that we assume that an endosomal escape takes place. In other words, a vesicle encloses the LNP and digests it via phospholipases, which are enzymes that digest phospholipids, in order to subsequently release the modRNA into the cytosol for translation. As you can see from this slide, we started to slowly understand more and more about the complexity and tight controlled regulation in 2015 at the latest. Originally, we assumed that only the RAS-related in-brain RAB GTPase proteins, of which we have identified 60 in human to date, as far as I know at last, regulated the endosomal lysosomal transition. This changed with increasing understanding of the peptidin cycle. And I hope I could already show what I mean when I talk about tightly regulated. The endosomal escape is one example of this absolutely tightly regulated set of cyclic rules. And this is precisely where the peptidin cycle and the membrane play an essential role even before the release of modRNA happens. From everything we know, which I will show in the third section, there is no doubt about the following fact. Cationic lipids, regardless of the complexity, in other words, as LNPs or pure cationic lipids or liposomes, completely shake up the anionic lipids of the membrane, pardon the untechnical term, and change the entire membrane structure. And they have also the potential to rupture large holes into the endosomes. The result is a catastrophe, because all receptors are functionally dependent on membrane integrity and load, and also act on it at the same time, which I won't only mention in this presentation and point out that I have also addressed this in my last 7 Substack articles, which can be seen as one huge Substack with more than 200 pages. Receptors such as the entire G protein coupled receptors, the vascular endothelial growth factor receptors, the epidermal growth factor receptors, and countless others, just to bring up some examples. So the change in the membrane will inevitably also send an initial cascade into the cell that comes into contact with these lipids of the lipid nanoparticles. And this is where it gets scary. Anyone who understands intracellular cascades knows that it is not the strength of the input signal, but the tightly regulated interactions, which is absolutely context dependent of the input signal with other signaling cascades. We call it crosstalks, cause some inhibit the strength of another signal, some activate or assemble another signal, etc., etc. In order to visualize the problem I want to draw attention to, I have labeled this really impressive slide, which shows the peptidin cycle, which is therefore to regulate endosomal lysosomal processes, membrane restructuring, and a lot of other processes. I wanted to point forward to the connected signal cascades of the mTOR mechanistic target of rapamycin complex 1, 2. Perhaps it should be mentioned at this point that one and the same protein can carry out various downstream and upstream interactions. Downstream means towards the nucleus, upstream, for example, feedback loops. And this can happen under certain conditions, and we always look exclusively at snapshots in such slides. And the slide discussed above should, at last I hope so, make everyone realize what an insane idea it would be to intervene directly in membrane regulation and disrupt this downstream network. What's more, it is much more complex than shown here. I will go into this a bit more detailed in the next section.